Okay, we have 22 participants today. Hopefully, we'll see more participants today. So at first, I would like to introduce myself. Next slide, please. I'm Mubashir Alam. I'm working at Center of Horticulture at Queensland Alliance for Agriculture and Food Innovation. So I'm a senior research fellow. So I have been working on macadamia and passion fruit breeding program. Next slide, please. At first, I'd like to acknowledge the country. The University of Queensland acknowledges the traditional owners and their custodianships of the lands on which we meet. We pay our respects to their ancestors and descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australian and global society. Next slide, please. Before introducing our new speakers, I would like to give you a little bit about the housekeeping things to mention. This seminar is scheduled for 12 until 1 p.m. At the conclusion of the seminar, we'll hold a question Q&A session. So if you have questions that you'd like answered, please use the Q&A button at the bottom of the screen, not the chat button, and you will address them at the conclusion of the seminar. Thank you. Next slide. Okay, so now I would like to introduce our speaker. Ms. Pragya Dhakal Poudel is a PhD candidate at the University of Queensland at Queensland Alliance for Agriculture and Food Innovation. And she's almost near the end of her PhD, so she submitted her thesis and waiting for the outcome from the examiners. Her work focuses on macadamia genetics and physiology, particularly the genetics and physiological factors influencing tree vigor. She has research interest in quantitative genetics, genome-assisted breeding techniques, and advanced breeding strategies to improve breeding outcomes. She's passionate about translating research into industry application, and she aims to support breeders and growers to optimize crop improvement and selection strategies. So now I'm going to hand over to our today's presentation, Pragya.
Good afternoon, everyone. Thank you for the introduction, Mo, and I would also like to thank the Coffee Science Seminar team for giving me this opportunity to present today. Today I'm talking about my PhD project, which is about the genetic and genomic insights into vigor in macadamia. My advisory team has Dr. Mubashir Alam as the principal advisor, Professor Rootstock, Dr. Lindsay Shaw, and Dr. Bibi Arif are my associate advisors. And I would also like to acknowledge the traditional owners and their custodianship of the lands on which we meet today. And I would like to pay my respect to their ancestors and their descendants. Okay, so to begin with, when we think of nuts that are native to Australia, one name stands out, the macadamia. Macadamias originated in Australia's rainforest around 60 million years ago, and today they are Australia's second largest nut export and the fourth largest horticultural export overall. In 2024, the Australian growers have produced nearly 54,000 tons of nut-in-shell macadamias, contributing to a global market that is projected to reach $7 billion by 2030. While macadamia production has expanded worldwide, led by South Africa, China, and Australia, this uniquely Australian crop is now enjoyed in over 40 countries. Macadamia belongs to the Protieaceae family and has 4 species. Most of the commercial cultivars are hybrids of Macadamia integrifolia and Macadamia tetraphylla, valued for their edible nuts. But Macadamia ternifolia and Macadamia jansseni they produce bitter and inedible nuts, and also remain largely unexplored for their potential benefits. Macadamia breeding in Australia is currently focused on selecting for industry targeted traits such as nut yield and quality, disease and pest resistance, and tree size or vigor. But breeding in tree crops like macadamia is difficult and time consuming. Current breeding cycle is at least 20 years due to different factors, including long juvenile period, which is at least 5 years before first nut production. And the peak production is after around 8 years of plantation. Also, large tree size at maturity means that a large amount of land is required for breeding purposes. There is primary emphasis on nut yield and quality, which has led to a slower prioritization of tree vigor traits in the breeding programs. And as a part of the National Macadamia Breeding Program, my PhD project focused on both tree vigor and strategies to reduce the lengthy selection cycle, contributing to more efficient macadamia breeding. Why is it important to prioritize vigor traits in macadamia improvement? Well, matured macadamia trees can grow up to 15 meters high and 10 meters wide, which produces a very large canopy. This leads to orchard crowding, shading, and the trees have reduced access to machines for pesticide application and also for different harvesting procedures. Low production efficiency, and also high labor and management cost. The high vigor also restricts orchard to a low density plantation, which is currently around 313 trees per hectare in many commercial orchards, while many other tree and nut crops have already shifted to a very high density plantation of more than 5,000 trees per hectare. In Australia, the high vigor in macadamia is often managed with several labor-intensive practices like machine hedging, heavy hand pruning, limb bending. But these practices require a lot of work, expensive equipment, which drives up the costs. Also, while hedging and pruning enhance light penetration, they also initially reduce yield and can also cause strong vegetative growth later on. Other tree crops, such as apple, citrus, and stone fruits like peach and cherries, have widely adopted dwarfing rootstocks to control tree vigor, to improve yield efficiency, and to facilitate high-density plantations. The breeding programs in these crops have developed yield-efficient and low-vigor cultivars. But in macadamia, the availability and research on dwarfing rootstocks is limited. Hatch to environment are the two commercially used rootstocks, but they offer minimal control over sign vigor. And there are some medium and low vigor cultivars like A16, A446, and A538, but highly efficient, high yield, low vigor cultivars are yet to be developed. So the overall objective of this project was to inform rootstock and sign breeding for vigor control in macadamia. I aimed to identify potential vigor controlling rootstocks, to investigate rootstock-mediated vigor control, to characterize the genetic architecture of vigor traits, to explore early vigor and precocity dynamics, and also to incorporate genomic-assisted breeding for vigor selection. So starting with the first study, where I evaluated several rootstock accessions for the potential for their potential vigor control abilities. So 24 different rootstock accessions were evaluated for their effects on early vigor traits of a common sign HGS 741. And the rootstock accessions consisted of 5 different categories, including wild germplasm, cultivars, potential dwarfs, elite selection from breeding programs, and the 2 commercial rootstocks hatched to environment. Also, the rootstocks were propagated as seedlings or cuttings. The vigor traits were measured from when the trees were 1 to 4 years old, and 6 traits were manually measured each year. And all the phenotypic data collected was analyzed using the REML mixed model analysis in GenStat. Firstly, I found a significant effect of rootstock on all vigor traits except for the total tree height. And there was also a significant difference between the 2 rootstock propagation method, that is seedling versus cuttings. This graph here, it shows the canopy volume of the HAES 741 cyan, which was grafted onto different cutting and seedling rootstocks. The red arrows, it shows the position of the commercial rootstock hatched to environment. So compared to the commercial rootstocks, there are several rootstocks such as the cuttings of Macadamia jansenii and BHY3, and also the seedlings of BDW3, A16, BHY4, and the Macadamia jansenii, which produced trees with smaller canopy volume. And the vigor-inducing rootstocks mostly included the selection from the breeding program with high yield and high selection index. The important thing to consider here is that this work was done on a very early age, and early vigor is crucial for orchard establishment, for stress tolerance, and also early yield in tree crops. Therefore, these early vigor promoting rootstocks should also be considered for further evaluation until maturity to see that if they exhibit controlled vigor at later stages, if they do control vigor at later stages, they could become ideal candidates for high density plantations, where these trees will combine the benefits of both early vigor and also manageable growth at maturity. And on the other hand, the rootstocks that reduced early vigor should also be carefully evaluated for yield efficiency. And if they show similar or better yield efficiency compared to the current standard rootstocks that are being used in the industry, they could be ideal candidates for high density plantations. But the continuous evaluation of rootstocks from both the ends until maturity is very important. In this study, we also estimated correlation between vigor traits of grafted trees and the same rootstock which was planted as an ungrafted tree. Correlations were strongly positive, especially for the canopy traits, which suggest that rootstock performance for canopy traits can be predicted from ungrafted trees, providing a practical approach to accelerated selection. And also low vigor cultivars are therefore strong candidates for vigor reducing rootstocks. Also, I looked at the genetic correlations between vigor traits of grafted trees and found that trunk circumference, which is generally used as an important indicator of growth and vigor, had a moderate to highly positive correlation with all other parameters, which indicates that this particular trait can also be used as an important representative of sign growth in macadamia, at least during the early growth stages. And therefore, the laborious phenotyping for multiple vigor traits could be replaced by a single measurement of trunk circumference. Moving on to study 2, so with the identification of rootstock effect on vigor from study 1, I was more interested in exploring whether rootstock affects the science stomatal traits, and also if there is any correlation between the stomatal traits and the rootstock mediated vigor. And for this, I phenotyped all the grafted trees from the rootstock trial for stomatal size and density using the traditional nail polish imprint method, where I obtained nail polish peels from nail polish peels with stomatal imprints from the cyan leaves, and then subjected them to imaging under microscope. The stomatal density was manually counted, while stomatal size was obtained using the image analysis tool called ImageJ. And all these data were analyzed using the REML mixed model analysis in HTML. And the analysis showed that rootstocks, the propagation type, that is cuttings versus seedlings, and the leaf position, which is the north and southern crown position within the tree, all had a significant effect on stomatal density, but not on stomatal size. And the total stomatal area was affected by the propagation type and the leaf position, but rootstocks did not have any influence on this trait. Stomatal morphology is involved in regulating a lot of different physiological processes in plants, including transpiration, water use efficiency, with an overall impact on growth and productivity. One of the genetic modifications for enhanced water use efficiency involves manipulating the surface area available for transpiration, which can either be achieved by reducing the size of individual stomata or by reducing the stomatal density. However, it is important to note that size and density are negatively correlated in many tree crops, including macadamia, where I found a negative correlation of 0.39 from this study. And in such case, moderate sized stomata in smaller density are preferred, as they are known to combine the benefits of reduced transpiration, better water use efficiency, but also maintaining photosynthesis without restricting the amount of water that is present for this process. And from my data, I have found certain rootstock candidates in like the BHY3 cuttings and the A268 seedlings, which showed lesser but moderate sized stomata, which may perform better under water restricted conditions. One of the objectives of this study was also to identify any association between stomatal and vigor traits. There was a small positive correlation of 0.29 between total stomatal area and canopy volume, which suggests that stomatal traits may be manipulated for vigor management. But I recommend for the study on gas exchanges to explore the physiology of vigor control by rootstocks, and also to explore the possible methods of modifying stomatal surface for vigor control. Working more on rootstocks, my third study explored the involvement of aquaporin genes in rootstock-mediated vigor control in macadamia. Aquaporins are actually a group of proteins that are present in the intracellular membrane and plasma membrane. And these group of genes facilitate water movement through plant cells. They are found to regulate vigor, growth rate, hydraulic conductivity, and photosynthesis in many horticultural crops. A recent study in grapes and olives have identified a significant role of aquaporins in the mechanism of vigor control by rootstocks. So in this study, I first used several bioinformatics tools to identify and characterize the aquaporin genes in the Macadamia integrifolia genome. since the whole genome sequence of the cultivar 741 is already available, I performed a blast analysis using the known aquaporin gene sequences of Arabidopsis and Populus. And from the bioinformatics analysis, I identified 37 putative aquaporin genes in the Macadamia integrifolia genome. These genes belong to 5 subfamilies named as plasma intrinsic proteins PIPs, tonoplast intrinsic proteins TIPs, not 26 like intrinsic proteins NIPs, the small and basic intrinsic proteins SIPs, and the unclassified intrinsic proteins, which are called XIPs. And the genes were named according to the phylogenetic relationship of aquaporin genes in macadamia with the aquaporins in Arabidopsis and Populus. I also looked at a whole lot of different information about all the 37 genes, including the chromosomal locations, the structure of each gene, and also motif analysis, which I'll be skipping for now. But the most important analysis is the conserved residue analysis that I did, where I looked at different amino acid residues that are present in the important locations of all these genes, and checked whether these genes that I identified had those particular amino acid composition in that location, which actually makes an aquaporin. For example, the NPA motif is one of the major locations in the aquaporin structure, which is responsible for the selectivity to water molecules. And I identified this particular combination of NPA, which is kind of an hallmark of being an aquaporin gene. So the identification of these NPA motif also supports the proper identification of aquaporin through my analysis. Once the aquaporin genes were identified, an expression analysis of 11 plasma intrinsic protein PIP genes, which was one of the subfamilies of aquaporin gene, was conducted. So I looked at the expression of these particular subfamily in dwarfing and vigorous rootstocks of macadamia. 4 dwarfing rootstocks with a single replicate and 3 vigorous rootstocks with 2 replicates were selected from the same rootstock trial. And I sampled leaf and stem tissues from from the selected grafted trees, and also the corresponding ungrafted trees. I extracted RNA from the leaf and stem tissues using a modified CTAB method, designed primers specific to the 11 PIP genes, synthesized cDNA, and then performed a real-time qPCR. These graphs here show the relative expression of PIP genes in leaf and stem tissues of vigorous and dwarfing rootstocks. Dwarfing rootstocks presented in red bars and vigorous in blue bars. And these comparisons are based only on the grafted trees, and the expression values were normalized to that of actin. Firstly, a few PIP genes, such as PIP14A and PIP28, had higher expressions compared to the other PIP genes. And interestingly, signs on dwarfing rootstocks had significantly higher expression of 9 out of 11 PIP genes in leaf tissues and 4 out of 11 PIP genes in stem tissues compared to the vigorous rootstocks. A similar comparison of vigorous and dwarfing rootstocks were also made on the corresponding ungrafted trees, and results were very similar. In leaf tissues, 7 out of 11 PIP genes showed higher expression in dwarfing rootstocks compared to the vigorous ones. And this observation suggests that the differential expression of these genes is an intrinsic characteristic of the rootstock genotype itself, rather than being influenced by the scion or the process of grafting. The expression levels of the PIP genes in leaf tissues of grafted and ungrafted trees were also correlated with 4 vigor traits, including total height, trunk circumference, canopy area, and canopy volume. And the correlations were significantly negative for most of the PIP genes, as highlighted in orange in this table. So I developed a possible hypothesis from all the results that I obtained from this study. Rootstock and scion water relations are highlighted to be one of the major mechanisms of vigor control in tree crops. The PIP genes are mostly overexpressed in stress conditions, particularly during water stress, to maintain water transport efficiency. The reduced water transport ability of dwarfing rootstocks, particularly due to the presence of lower root biomass or smaller xylem vessels, has been identified in many tree crops. And based on the studies in other tree crops and the results from the current study, it can be hypothesized that the elevated expression of PIP genes in dwarfing rootstocks would be a compensatory mechanism to maintain water transport efficiency despite the reduced water transport ability of dwarfing rootstocks. It also hints toward improved resilience and performance of dwarfing rootstocks especially under challenging environmental conditions. But further research involving detailed analysis of the root biomass, vessel structure, and photosynthetic characteristics is required. Moving away from rootstocks, I also focused on selecting low vigor cultivars, and this study investigated the genetic parameters of vigor traits in a breeding population of macadamia, where I looked at the genetic variation, heritability, and correlations among different ages for vigor traits, and also applied a genomic prediction model to predict vigor. This study was conducted on a trial within an Australian macadamia breeding program population located at the Bundaberg Research Facility, and which was planted in 2013. The population had 838 progenies from 57 families, and all these trees were phenotyped for 5 vigor traits, manually measured from 2 to 10 years after plantation. And all the phenotypic data I obtained was analyzed using the mixed model analysis in HMNR. I looked at the genetic variants of different vigor traits. However, I will only be presenting 2 important traits, height and trunk circumference today. The interesting information here is how the additive genetic variants and also the narrow sense heritability of these traits increased from age 2 until age 7, but somewhat stabilized from around age 7 to age 10. This likely marks the full expression of genetic potential for growth traits by age 7. And also the phenotypic and genetic correlations between vigor at age 7 and age 10 were higher than 0.8 for both tree height and trunk circumference. And for breeding purposes, the information from combined information from variance component, heritability, and correlation suggests that selection for vigor can be effectively done at around age 7, as the selected individuals are likely to maintain their relative size through age 10. I also estimated breeding values of different parent cultivars in this study and identified 10 parent cultivars with low breeding values for vigor, which are potential candidates for developing progenies with a compact tree size. And similarly, I created a dendrogram with heat map based on the breeding values of all the parent cultivars and compared the vigor pattern across different growth stages. And this particular cluster, cluster 3, it highlighted several parents which showed high growth at early ages, which is until age 7, but the growth rate was slowed down after age 7. And as I mentioned before, early vigor is crucial for crop establishment and early yield. And hence, these parents from this particular cluster with balanced vigor are promising candidates for combining rapid establishment and also with manageable growth at maturity. And then I applied a GBLOB genomic prediction model to a training population of 299 seedling progenies for 4 vigor traits at age 7, and then tested this model to predict vigor of another population with 183 seedling progenies. And despite having a limited marker size of around 2,600 SNPs post-filtering, this model showed a prediction accuracy which was greater than 0.61, demonstrating the potential of genomic prediction to enhance selection efficiency for vigor traits in breeding programs. Finally, the last study explored the association between early vigor and precocity in macadamia, where I estimated correlations between these traits and then performed a genome-wide association study to identify any common markers governing both the traits. This study used historical phenotypic data from 904 seedling progenies planted across 6 different sites in Queensland and New South Wales. Early vigor is represented by total height, canopy width, and canopy volume, and precocity represented by total nut-in-shell mass and cumulative nut-in-shell mass, all at age 5. And also 219 progenies from this big population were genotyped using the diversity arrays technology platform, which gave us 12,732 SNP markers. And the phenotypic and genotypic data were combined to perform a genome-wide association study using the mixed linear model in the Tassel software. But first, I looked at the correlations between early vigor and precocity, which was moderate and positive, with the highest correlation observed between canopy width and total nut-in-shell mass, with a phenotypic correlation of 0.42 and a genetic correlation of 0.29. The results from the genome-wide association study then identified 2 significant markers for height, 5 for canopy width, 4 for canopy volume, 2 for total nut-in-shell mass, and 5 for cumulative nut-in-shell mass. And one of the significant markers was common between height and cumulative nut-in-shell mass, and another was common between height, canopy volume, total and cumulative nut-in-shell mass. I also looked at genes that were closely localized to these specific markers, and they were functionally annotated for growth, development, and pathogen difference in other plant species. However, I would like to make a note here that this second common marker 10079 had a very low minor allele frequency of 0.007, which makes this a rare allele. And the instances of rare marker being a causal marker, especially in growth and yield of plant species, is evidential. And therefore, when we are excluding low minor allele frequency SNPs by filtering, we are preventing the identification of these important alleles, which made me include these low minor allele frequency markers to see if they have any significant influence on the traits of my interest. To further understand the effect of different alleles of these markers, I visualized the allelic effect on each phenotype. The rare heterozygous allele AC of this marker showed a favorable effect on precocity, but it had its effect on early vigor was negligible. So selecting for this heterozygote could aid in increased precocity, but also without adversely affecting the early vigor. But due to the rare presence of this allele, results should be interpreted very carefully, and validation of this marker in preferably larger population before utilizing it in marker-assisted selection is very important. And similarly, at the other common marker 4004, despite being rare, the heterozygous allele CG had a highly favorable impact on precocity while also reducing early vigor. So selecting accessions for this rare allele might be beneficial in identifying cultivars suitable for high-density planting systems, promoting small tree size, but also with high precocity. Once the candidate markers are verified, the markers can be efficiently used for future marker-assisted selection to select for early yielding cultivars with required early vigor. And the marker-assisted selection will replace the tedious phenotyping and evaluation stage of the breeding cycle, as it allows pre-screening for traits at seedling stage. This potentially reduces selection cycle by 8 to 10 years, and also utilizing common markers by selecting for 2 traits using a single marker makes the selection process time and cost effective, and also allows higher genetic gain in breeding programs. So I'd like to end the project description with the practical gains and outcomes from this project that are relevant to the macadamia industry. Firstly, the information are directly relevant to both the growers and plant breeders within the industry. Outcomes from the first study provides breeders and growers with information on different rootstocks that have the possibilities of both reducing and inducing early vigor. And the interesting rootstocks for potential users, dwarfing rootstocks, can be selected for future breeding. And the possibility of vigor prediction of grafted trees through the analysis of ungrafted trees potentially shortens the time required for rootstock evaluation by eliminating the process of grafting for rootstock evaluation purposes. And the findings from the second study are particularly relevant in understanding how rootstock selection can influence not just growth, but also physiological and morphological traits such as stomatal density of the scion. This highlights the possibility of selecting rootstocks for water use efficiency and stress resilience in macadamia. And these insights can guide both growers and breeders in making more informed decisions about rootstock choice and orchard management strategies. The third study provides the protein sequences of putative aquaporin genes in macadamia, which can be used by researchers for further functional research of these genes. And this chapter also provides a basis of a foundation of physiological and molecular mechanism of vigor control by rootstocks. And the possible hypothesis that is generated from the findings in this study is an important avenue for further research in this area. The fourth study provides a comprehensive data set that can guide breeding for vigor traits. Selecting individuals at age 7 is a sound strategy due to the high heritability of traits at this age, and also because of their strong correlation with performance at age 10. This approach also ensures that selection decisions are both effective and predictive of long-term success, making age 7 as an optimal point for advancing breeding program. Also, a high correlation of trunk circumference with other vigor traits observed in both the studies 1 and 4 suggest that this parameter could be used as an indicator of overall tree vigor, and therefore, as I mentioned before, the laborious phenotyping for numerous vigor traits could be replaced by phenotyping for this single trait. And the genomic prediction model used in this study can be used in breeding programs to enhance genetic gain, but strategies strategies like improving the number of SNP markers and increasing the population could help in achieving higher prediction accuracies as well. And finally, the last study provides important dynamics of early vigor and precocity relationships. A positive correlation and common SNP marker between these two traits shows the importance of early vigor for precocity. And the significant SNP markers identified here can be used for marker-assisted selection, especially the common marker associated with both the traits, which allows selecting for two traits at the same time, promoting accelerated selection. But of course, validation of these markers is required before utilizing them in marker-assisted selection. Okay. I have a big list of people and institute that I need to acknowledge. Firstly, my principal advisor, who has been a great support throughout this journey, and also my advisory team for their ideas, immense support and encouragement. Dr. Joanne D. Fabry, my former advisor, for her contributions in statistical analysis. Dr. Max Cowan for his help with data collection, sampling, and writing. The Progress Review Panel for their time and advice, the University of Queensland and QAAFI for providing the scholarship and required resources for this project. Port Innovation for funding the project my research is part of. Dr. Burhan Ahmed for helping me with the bioinformatic analysis. Palak, Jean, Sam, Rod, Chong, and Jung for all their help with numerous data collection and field work. And the macadamia research team and all other staff members at the Modulation Research Facility. Thank you so much for listening, and I'm happy to answer any questions. And I would like to hand it over back to Mo to facilitate the Q&A session. Thank you.
Excellent, Pragya. That's a nice presentation. So now the floor is open for all to answer any questions. So if you want to ask any questions, please use Q&A tab, not the chat, please. So we haven't received any yet. Just in the meantime, I'd like to ask a few questions. So yeah, you highlighted a few industry outcomes from your project. So do you think you can select any rootstocks for our industry from your research?
Yeah, just like how I presented earlier in my results from the rootstock trial, we have a few candidate accessions. One of the accession is a selection from the National Breeding Program, which is a selection with high yield index. So that can be a potential rootstock for further evaluation. And also the macadamia jansenii, which showed vigor controlling ability when propagated either as cutting or seedling. That also has a very good possibility of being used as dwarfing rootstock here, but further research is obviously needed with a lot of replicates.
So that Jansenine is a cutting or seedling?
So both, we had the Jansenii propagated as both cuttings and seedlings.
And both reduce the size.
Yes, both reduce the size significantly.
So in that case, we can use seedling as a rootstock. We don't have to worry about propagating using tissue culture.
Yes.
Okay, good. That's fantastic. Yeah. Yep. So we haven't received any questions from our tab, Q&A tab. So another thing is the genomic prediction. So your accuracy is 0.5, am I right? Something?
Higher than 0.6.
Higher than 0.6. So do you think it is enough for selecting accessions?
This study is kind of a very preliminary genomic prediction that we did, but there is a lot of scope for increasing that prediction accuracy, especially if we are able to, if we use a larger SNP larger number of SNP data, and also if we can use a bigger population, then there is a very good scope of increasing the prediction accuracy. And then the more the prediction accuracy, I think the better the model will be.
But you said heritability heritable for vigor is high, am I right?
Yes.
Oh, that's good.
Too high. Yes.
Okay, that's good. Good, fantastic. So that's good. So we haven't received any questions. So I think you did present nicely, so everyone could understand what is said. So we'll wait for a few more minutes. So we'll see if anyone asks any question in the Q&A, using Q&A button. Another thing is you have chosen aquaporin genes, gene expression. You looked at the aquaporin gene expression. So why did you select aquaporin genes, not any other genes, or to look at the molecular mechanism of rootstock-mediated vigor control?
The main reason that I used aquaporin gene is there are a different different hypotheses that have been generated over years that is related to the process of rootstock mediated vigor. But the most widely studied one has been the rootstock and sign water relations, where it has been showed that dwarfing rootstocks are usually transporting lesser amount of water to the sign, which is how the vigor is being controlled. And aquaporins are the major proteins or genes that are present in tree crops, which regulate the water transportation. Either it is a cell-to-cell water transportation, that is a short distance water transportation, or a long distance water transportation, which is from rootstock to sign, which is why I was more interested in looking at this particular set of genes. And also I selected this particular subfamily called PI, plasma intrinsic protein genes, because that particular gene is more involved in long distance water transport compared to the other subfamilies, which such as the DIP subfamily, which is more involved in short distance water transport.
Yeah. Good. So you looked at the expression profile in the leaf or in the root?
In the leaf and stem. I was very interested in looking at the roots as well, and I trialed a few different RNA extraction methods for the root tissues, but I was not able to get a very good quality RNA from the root tissues, which is why I had to move on with leaf and stem. But looking at the expression in root is very important.
Good. Well done. So do you think we need to stop our research on rootstock now, or just we need to continue? We need to do more. Do you have any plan? Do you have any plan?
We definitely need to do a lot, especially through my literature review, I have had a lot of different ideas, such as when I'm talking about the hypothesis that I generated from my aquaporin study, how the dwarfing rootstocks are known to have a smaller root biomass, smaller vessel size from previous studies. And if we can do the similar channel vessel study or root biomass study in rootstocks, and also look at different gas exchanges in the sign, like photosynthesis or chlorophyll, or those sort of water exchange parameters, that would be
Good,
great.
good, good. So we haven't received any more questions using Q&A tab. So, but I think you did very well. So,
Thank you
but
so much.
I would like to thank everyone who attended today's science seminar. If you enjoyed today's seminar, please do the poll at the end. And if you haven't already signed up to the seminar invitation list, please do at coffee.eq.edu.au/science_seminars. And you can use this barcode to sign up yourself. Thank you, everyone. Bye.
Thank you so much. Bye.